Thank you very much for the introduction. And it's very good to be included in this session, and it definitely makes me feel very youthful because now I'm apparently a next generation scientist, so it's great. Thank you for the invitation as well. I'm a virologist, as you know, and I'll work on foodborne viruses. And so I might actually go to some introduction about viruses. I apologize in advance if you know about them, but I just thought that they deserve a little bit of introduction. So foodborne transmission of human enteric viruses is a well-recognized, widespread public health concern. So norovirus and hepatitis A virus are the major causes of non-bacterial foodborne illnesses. And what's important about them is that they are highly infectious and very resistant in the environment. So usually when we talk about norovirus and hepatitis A virus, you would assume either shellfish consumption or fresh produce consumption. But we recently had some outbreaks that were associated with low moisture food, and I'm going to talk about them in this presentation. So norovirus, just a little bit of introduction. It's a single-stranded RNA virus with positive polarity. It's non-enveloped. And so it causes a disease that usually lasts 2 to 3 days. Maybe some of us have first-hand experience with it. It feels really awful. But it usually resolves by itself if you're not immunocompromised. So it's genetically very heterogeneous. So it's divided into several genogroups, and then each genogroup is further subdivided into genotypes. So, and these classifications are, of course, based on genetic diversity. Right now, unfortunately, there is no robust and readily available cell culture system for norovirus. So what we experience and what we use for applied works is using the surrogates of norovirus, which is murine norovirus, which we will refer to MNV from now on, and feline calicivirus, which is FCV, the acronym. And so a lot of applied works have been used, these surrogates. Hepatitis A virus, it causes acute hepatitis. It usually lasts less than 2 months, again, if you're not immunocompromised. There is an effective vaccine against that. However, outbreaks still occur, and it causes morbidity throughout the world. It is not as genetically heterogeneous as norovirus. It's classified into 6 genotypes. Genotype 1 to 3 infect humans. And what's important about Hep A is that it only has 1 serotype. So that makes producing a vaccine against it easier, and working with this virus also is a little bit easier because of that. So I'll explain why. So why do we study low moisture foods in terms of virus survival and activation? First of all, foodborne viruses are very resistant in the environment. They can survive for long term just in any conditions, including low moisture conditions. And they're generally considered ready-to-eat products. So normally this pasteurization or any processing that would reduce the pathogen load in them is not applied on low moisture food. And as I mentioned, recently there were several outbreaks associated with low moisture foods and norovirus or hepatitis A virus. For example, there was this very large outbreak in Japan and South Korea associated with dried seaweed that is called nori. And it caused over 2,000 people being sick from norovirus. And more recently in Europe, there is an outbreak of hepatitis A infection associated with imported dates. So that shows that viruses can be transmitted through low moisture foods, but unfortunately there is not much we know about how they're transmitted. because we haven't tested methodologies so far to be able to extract these viruses from these commodities and then study them. So the objective of this project was to, first of all, develop method or implement already existing methods for isolation and enumeration of foodborne viruses from low moisture foods. And then after that, using those methods for examining survival and then inactivation of viruses in these commodities. And also we explored the possibility of using some next generation sequencing methodology to study the possible mutations or the variations that exist in the virus populations that can be in low moisture foods. So just overview of the methodology that we used in this work. So as you know, we used pistachio, cornflakes as a breakfast cereal, and chocolate liqueur. We inoculate them with hepatitis A virus, feline calicivirus, and murine norovirus, and incubated at room temperature for 1 day up to 4 weeks. And we compared two methods. One method was the ISO 15216 method, which is an accepted method, worldwide validated for extraction of foodborne viruses from different commodities, but it hasn't been validated for implication in low moisture foods. And this method is basically based on precipitation of the virus and then further characterization. So you can do quantitation by either droplet digital PCR or plaque assay. On the other hand, we also use magnetic bead assays to be able to extract viruses and then quantitate by droplet digital PCR. So just a little bit about the HAV capture by the beads. As I mentioned, there is one serotype for hepatitis A virus. So it's easy to code magnetic beads with anti-Hep A antibody and use those magnetic beads to capture hepatitis A virus in different food commodities. However, as I mentioned, norovirus is very heterogeneous, and like in terms of antigenicity, you cannot use antibodies to capture norovirus because the antibody against one strain might not work against other strains or other genotypes. So for that, we use the porcine gastric mucin, which has been, this method has been used and developed by David Kingsley, who's also in the room. So it's been a very successful method for capturing norovirus and surrogate from different food commodities or even from clinical samples. So this molecule resembles one of the receptors for the virus, and then therefore it can bind to the virus, and then therefore you can use it to capture. And as you see, it's mucin, so it's basically a glycoprotein that has some carbohydrate chains. So now we move on to study the recovery rates using the both methods, just comparing ISO 15216 method with the bead-based method in recovery of different viruses from the studied commodities. So as you see, the bead-based method could give us very good recovery compared to the traditional ISO method, ISO 15216 method. And so we got good recoveries in chocolate, pistachio for both FCV and MNV. However, when we looked at cornflakes, we could not recover any of the virus, either FCV or MNV, by the bead-based assay. So at this point, we were coming with hypotheses that maybe the high carbohydrate content of cornflakes outcompeted the beads for binding to the virus in that sense. And so this is a hypothesis that needs further exploration. However, when you use hepatitis A virus, and as I mentioned, there is no carbohydrate involved. So for cornflakes, it's just basically based on protein-protein interaction or antibody-antigen interaction. We didn't have that problem, and the bead-based method continued to give us higher recovery rates compared to the ISO method, which is based on precipitation of the virus. So we looked at the virus survival. First virus is FCV, the first surrogate for norovirus. And so for survival up to 4 weeks, by both bead-based method and ISO method, they basically gave us similar results. And after 4 weeks incubation at room temperature, we get less than, definitely less than 1 log reduction in infectivity. So in the virus that we could, in the nucleic acid of the virus that we could detect. And then similar results could be obtained by plaque assay, which is basically an infectivity assay. So now you're looking at the infectious virus and counting plaques. And again, for FCV, after 4 weeks incubation in low moisture food, basically the reduction in the viral infectivity is around 1 log, so it's not that high. MNV, similar story as FCV in pistachio and chocolate and cornflakes. 4 weeks incubation time gave us in the neighborhood of like half a log to like definitely less than 1 log reduction. And the plaque assay and the infectivity assay just gave us the reduction in infectivity around 1 log. So this is an ongoing system and work in progress project. So we are still trying to complete this puzzle. Like there are a few pieces left here. So we are still examining the 4 weeks titer in cornflakes, but in general, we know that we should expect something around less than 1.5 log reduction. Hepatitis A virus, very similar story, just the genome of the virus by bead-based assay or ISO method, it stays around between half a log to 1 log reduction after 4 weeks incubation. We are still working on the plaque assay, but it seems that the reduction, at least for the pistachio that we have the complete picture for now, it's around 1.5 log. Next, we use metagenomics approach to look at the genetics of the population of the viruses that we could retrieve after 4 weeks incubation in different low moisture foods. And for that, we use a metagenomic approach, which is basically, we didn't use any amplicon-based approach. It's rather unbiased. And we basically did RNA-seq on the food commodities that were inoculated from time 0 to week 4. And then we could assemble the full genome of the viruses that way. So I understand that it's a very busy slide. We actually, from some of the viruses, specifically for FCV, we observed quite a bit of change from time 0 to week 4. So some of these viruses, some of these changes are coding, some of them are not. And then the other thing that I just want to draw your attention to quickly is the ratio, because we did deep sequencing, so we could look into the deep population of the viruses that were in those food samples. And then, as you also know, and it's always mentioned in the textbooks, that viruses exist in a swarm of mutations that's usually people like to call quasi-species or just varied population. There is for some of those nucleic acid changes, we could see that the SNPs or single nucleotide polymorphisms were not that clean. So there was just a mixture of genetic variations that exist in those populations, but some of them were dominant. So when you're calling SNP, or even you're calling it variation, it depends on the coverage that you get. Sometimes those nucleotide changes can be obvious that they're actually a heterogeneous population that you're working with. Next, we actually partnered with industry. They made us a unit. It's called Advanced Oxidative Process Unit. What it does is that it uses ozone, UVC, and hydrogen peroxide vapor all at the same time to treat foods. So this is rather a large unit. It has a conveyor belt, and you put your sample in there, and you can vary the treatment time from 0 seconds, obviously, untreated, or 30 seconds, or 60 seconds, or 1 minute, and look at the level of inactivation. So we looked at the level of inactivation both by plaque assay and droplet digital PCR. So we compared them head to head. And as you see, like, it depends on the food commodity that you are working with. For example, chocolate, which had, as I showed you on our very first slide, it has a very flat surface, so it can get the treatment easier. The inactivation reached to 100%. So we didn't even see any activity after. But again, for other food commodities that have complicated surfaces, like pistachio and cornflakes, again, as the surface gets more complicated, the level of inactivation decreases. This is the data for FCV. For MNV, again, similar story. For a chocolate plaque assay, we got 100% inactivation. Again, for pistachios and cornflakes, the level of inactivation was lower. The other thing that I wanted to draw your attention to is that you can see there is a difference between the data that we get by plaque assay than the data that we get by just looking at the nucleic acid level. And that shows you that usually detecting the nucleic acid by itself doesn't mean that the virus that you detect is infectious or is able to replicate. So that means that even in food samples, when you're detecting the genome of the virus, it's very difficult to interpret whether the virus is infectious or not. Next, we looked at the HIV inactivation by that process. And so far, we are working on the plaque assay results, so I don't have the results to show you, but a similar observation with chocolate. We got good results in terms of inactivation, but so far, we also got impressive results for cornflakes for Hep A. So we are actually waiting to get the plaque assay results to complete that story. So in conclusion, so we showed that we can use the magnetic bead-based assay to isolate viruses. And we could show that basically magnetic bead assay is more convenient to work with. It's faster, but at the same time, it might not be efficient in capturing the virus depending on your food commodity. As I mentioned, for cornflakes, we could not retrieve the virus using the bead-based assay, but for hepatitis A, we had a good recovery in cornflakes or in all commodities that we looked into. So in general, it seems that all these viruses that we studied have very persistent, both in terms of their genetic material and in terms of maintaining their infectivity, at least during the time that we were looking at 4 weeks in infection. 4 weeks incubation. So just incubation by itself at room temperature doesn't cause a significant decrease in the viral load, or doesn't cause inactivation of the virus by itself. So in terms of infectivity reduction, we just saw between 1 to 1.5 log reduction after 4 weeks incubation. And then depending on the commodity that we used, for example, for chocolate, that the surface was perfectly flat, we could get 100% reduction using the AOP. And then we also used some next generation sequencing and stem cell analysis, and we identified that although the viruses don't replicate on foods, but the incubation at room temperature might actually lead to the survival of certain variations within a population that could outcompete or could be potentially more fit for transmission on low moisture foods. And at the end, this was a huge project. It wouldn't be possible without the help of my students and my technician. And then I also wanted to definitely thank ILSI for funding the project and made it possible for us to work on this interesting project. Thank you.
Are there any questions for Neda?
Very good talk.
Thank you.
With the advanced oxidative process, obviously it's very reliant on the kind of UV dose hydrogen peroxide and ozone content.
Yeah.
When we did it with Salmonella on cornflakes, we needed a certain 5%. We still got a 5 log reduction. Did you actually optimize the AOP process, or did you use a set treatment?
So it's a very good question. However, like the only thing that we actually played with was the treatment time. So the concentration of hydrogen peroxide vapor remained the same, and for ozone and UVC, everything remained the same. So we didn't actually vary these parameters.
Yeah, you could see maybe the kill rate might be even higher if it was, but thanks for that.
Yeah, so like it would be also interesting to look into the effect of individual treatment, but there is a wealth of data in the literature for each of these inactivation mechanisms for surrogates of norovirus, even hepatitis A virus. But AOP unit just gave us the opportunity to work with all these treatments all at the same time.
Hi, Neda. Jeff Farber, University of Guelph. Very nice talk. Neda, just wanted to get your opinion, what you think. I mean, you saw about a 1 to 1.5 reduction in infectivity for over 4 weeks. Do you think if what has been stored, let's say for 2, 3, 4 months, you would have seen further reduction in infectivity?
Very good point. Well, I can't say because we haven't looked into it, so it's difficult to predict the behavior of the virus. But in general, like as you see, like even it depends how much virus you start with. So like what would be your contamination at the beginning. So this is, of course, artificially contaminated commodities. And so we started with very high loads of the virus, so 10 to the 7, 10 to the 6. So the viral load at the beginning was very high. But in natural contamination, again, the viral load is usually much, much lower. So again, if you start again with 10 to the 1, 10 to the 2 of the virus load, you might actually eventually get some.
Yeah, that's why I'm wondering if companies could use that as a strategy, hold something for 3 or 4 months and then get the viral load infectivity down to 0.
Sure, and again, there are studies, not on low moisture foods, but on surfaces that they actually looked into the effect of just incubation time and room temperature for virus inactivation. And so again, the number is like, it's not something that's mind-blowingly low, but at natural contamination level, it might be meaningful.